Hello again, biologists. Mr. Kinnear here with another video lesson for National 5 Biology. We're moving on to Unit 2, Multicellular Organisms. So we've obviously finished Unit 1, Cell Biology, where we just looked at individual cells and how they perhaps interacted. We're now getting a bit more complex and grouping them together in multicellular organisms. So we'll start here today, Key Area 2.1, producing new cells. We'll then talk about how control and communication happens within multicellular organisms, reproduction, and then variation and inheritance. So why is there differences between organisms in the same species and how that information is passed down from parents, grandparents, etc. We'll look at transport systems in both plants and animals, so how they transport different molecules around their bodies, and also absorption of materials, which will look at digestive system and the lungs mainly. DNA is the starting point for us producing new cells, and obviously we've talked about that in cell biology. Most cells contain somatic cells, so the body cells, and gametes, sex cells, so in humans that would be sperm and egg. Now we should know that there is different DNA complement within those. Body cells normally contain two sets of chromosomes in their nucleus. In humans, as you can see from this diagram, it's two sets of 23, so 46 chromosomes in total. We'll also know that the sex cells, that are sperm and egg in terms of humans, contain half of that genetic information. So every chromosome in one set has a matching partner in the other set with the same length of DNA and the same genes. So that will be really important when we look at reproduction. It will be really important when it comes to variation and inheritance as well. Cells that contain two sets of chromosomes are said to be diploid. And again, that's a word that you will need to know as we go forward. So when we're thinking about producing new cells, that comes about through cell division. All cells are produced by the division of other cells, so one cell becoming two. Multicellular organisms themselves start their lives as a single cell. That shouldn't be news to you, a fertilized egg. And we get that from a sperm and an egg in humans and mammals. Gametes have only one set of chromosomes, are said to be haploid. So like in the previous slide, we talked about diploid and humans being 46. Our haploid egg and our haploid sperm contain 23 chromosomes each, one set of chromosomes each. And obviously during the process of fertilization, we end up with a diploid zygote, a fertilized egg, which contains 46 chromosomes. So half of that's come from mum, half has come from dad. Cell division allows for growth and to replace damaged tissue. So obviously that zygote divides and becomes two cells, two become four, etc., etc., till we get to a fetus, a baby, a child. And obviously as that child grows, then they again go through the process of cell division, and they all are identical to the parent cell. Then equally, as we get to adulthood, we still have to repair our body. So if it's a cut or it's damaged or anything like that, then we're repairing that, or a break in the arm. We have to grow new cells, bone cells or skin cells, etc. And that happens again through the process of cell division, producing identical daughter cells to the parent cell each time. So cell division is the process of producing new cells for growth and repair. Mitosis is the process of dividing the nucleus during cell division. The first step of that is to replicate the chromosomes that are in the nucleus. This makes sure that the diploid chromosome complement of cells is maintained and that every single daughter cell has all the genes needed to function correctly. And that's going to be really important for the proper functioning of this multicellular organism, which these daughter cells are going to be part of. So we'll take you through this process of mitosis stage by stage, 6 stages in total. The first one is when we can start to see the DNA become visible after DNA replication. As I said, the first step is to make sure that our DNA is replicated. If we're going to split this cell into two, we need to make sure that both of those cells at the end have all the DNA, the full diploid complement. So we're going to replicate that DNA before we do anything. Now DNA is quite fascinating in that it is a tiny molecule because it can fit into the nucleus of every single one of your cells, but equally huge because it can carry all the information required to make you, you. If you were to take it out of one cell and stretch it from point to point, then it'd be about 1.8 to 2 meters long. So that's the average height of an adult male. So huge in that sense, but equally coiled and folded into each and every single one of the nuclei of our cells. In stage 2, the chromosomes become shortened. They appear as chromatids. They're joined by a centromere. Now, in this example, we've got 4 chromosomes. Obviously, we have a lot more in the human body. We've got 23 pairs. But in this instance, we've got just 4 to show the process simply. The centromere is this dot in the middle, and what you can see is each of the chromosomes is now made up of 2 chromatids, and that's the same for each of those 4 chromosomes. So again, this process can be seen under microscopy in the process of mitosis. In stage 3, the nuclear membrane disappears, so the DNA is normally held within the nucleus, which has a membrane, but that disappears, and we can start to see the chromosomes lined up on the equator. And just like we have on the Earth, we have an imaginary line in the middle. We have a North Pole at the top, we have a South Pole at the bottom, and an equator, an imaginary line. And faintly, you might see these things called spindle fibers. They're attaching onto the centromere of each of the chromosomes. They're going to employ an important job in stage 4. So as you can see, in stage 4, our chromatids are being pulled apart. So each side is getting the same amount of DNA pulled to it by these spindle fibers. Effectively, they are shortening and pulling the chromosomes apart, pulling a chromatid to each pole. So in stage 5, you can start to see that a nuclear membrane is forming around the chromosomes at each pole. The good news is that, as you can see, we've got 2 red and 2 black chromosomes at each side. Now we're starting to see the cytoplasm divide. So again, a new membrane is forming to make 2 daughter cells. So Bob's your uncle, in stage 6, we now have 2 identical daughter cells formed, both with their own cell membrane and nuclear membrane. Again, the DNA doesn't need to be condensed for this process, so it now will disappear from sight. So here we have all the stages together, obviously starting here at stage 1, moving all the way around to stage 6 with our 2 identical daughter cells. Now, it doesn't move immediately from stage 6 back into stage 1. There will be some pause in that process, but this is the same process that can happen in bacterial cells. So for instance, if you eat something dodgy or don't wash your hands before eating and after going to the toilet, you can ingest a huge amount of bacteria, say for instance 1,000. Well, that 1,000 can become 2,000 in as little as 20 to 30 minutes, and 2,000 could be 4,000 within an hour, and 64,000 within just a few hours. So that's how you can get quite unwell very quickly from bacteria, because they replicate through this process of mitosis. One cell can become a huge amount of cells in a very short period of time. So that's one of the reasons why you need to be careful. But the good thing for bacteria in that process is that all of those cells are identical, containing the same genetic information to start off with. And obviously that's the same good benefit for you when you're replicating your cells with growth and repair, that each of those cells will have the same genetic information from the parent cell to the daughter cell. Now, the cells that we were looking at in the process of cell division and mitosis looked fairly ordinary animal cells with a cell membrane, cytoplasm, and a nucleus. However, in multicellular organisms, those cells become specialized. They need to be carrying out different functions, so therefore they look different and act differently. That allows the organism to function more efficiently. Those specialized cells in animals develop from stem cells. And stem cells are unspecialized cells. So again, like those cells we saw in the process of mitosis, fairly ordinary. And they can do 2 things: self-renew through the process of mitosis, or differentiate into a specialised cell. So they can come from unspecialised into specialised through the process of differentiation. So like I said, a stem cell, fairly ordinary looking animal cell. It can do two things: copy itself or self-renew to create another stem cell, or it can differentiate, so specialise. For example, a muscle cell or a nerve cell. Now that isn't a particularly good diagram of a specialised cell, but you can see from it that the specialised cell looks different from a stem cell. It carries all the same genetic information as the stem cell does, but only some of that genetic information is actually turned on so that it can specialise into, as I say, muscle cell or nerve cell or any of the other specialised cells we're going to look at. There are two main types of stem cells that we're interested in National 5 biology, the first being embryonic stem cells. As the name suggests, they can be found in early embryos and they can develop into any type of body cell. So once we have fertilisation, a sperm meeting an egg successfully, we've got 46 chromosomes in there now within a human, and that cell will spend its first few days replicating until it becomes a rather large ball of cells. No specialisation in there yet, so purely a large ball of cells, and that's where we get our embryonic stem cells. A second type is tissue stem cells, and they can be found in all grown adults and children. So we have specialisation within our bodies, within our organs, and there are specialised stem cells in there that can develop into a limited types of cells. So we'll talk about them as we go forth. So embryonic stem cells, as you can see, a bundle of cells there. That's what you'd have after 4 or 5 days worth of self-renewal within the uterus. Then we have differentiation. So within the fetus, those will develop into muscle cells, nerve cells, red blood cells, white blood cells, etc. Those are known as pluripotent. They can become any cell in the body. They are, however, a little bit more specialised, but as far as we're concerned, any cell type in the body, and they are known as pluripotent. In some instances, tissue stem cells can be referred to as adult stem cells. It was originally thought that these cells were only found in adults, and there used to be phrases such as child stem cells. But what we found is that these tissue stem cells are found in places like the surface of the eye, brain, skin, the breast, intestines, testicles, muscles, bone marrow, etc. So various places around the body with some form of specialisation, allowing us to renew and repair different parts of our body. Tissue stem cells can often make several kinds of specialised cells, but they are more limited than embryonic. Tissue stem cells can only make the kinds of cells found in the tissue they belong to. So in this example, blood stem cells can only make the different kinds of cells found in the blood. So for instance, red blood cells, white blood cells, and platelets. Nervous stem cells can make the varieties of different nerve cells. Muscle stem cells obviously can make muscles. Therefore, they're referred to as multipotent. They can produce multiple types of cells, but not all. So that word multipotent and pluripotent, you need to make sure you understand those two words and the differences between them and which types of stem cells they apply to. You may be asked for some medical or therapeutic uses of stem cells. So these are a couple to have in your locker to use as and when you need them. First one being skin grafts. Now we need to be careful here. Traditional skin grafts are taken from people's back or leg to put some skin on burns which are more prominent. So somebody's face, if you've got a burn in your face, you might be given a skin graft to replace the skin on your face. However, if we're talking about stem cells, we're actually growing it in a lab. So we take some skin stem cells and we grow a layer of skin in the lab which can then be grafted on to the burn victim. Second one is bone marrow. So again, for treating leukemia. So bone marrow is where blood stem cells reside. So if anyone is suffering from leukemia and treated for that, they might need a bone marrow transplant. So it's replacing blood stem cells. Again, it has to be specific. So you have to go through some tests to make sure that you're a suitable donor for that person. But you get some bone marrow donation, and that then helps regenerate blood cells in the body. And the last one is still in the process of being tested. So growing heart muscle. So if it has been damaged after a heart attack or heart disease, then you can actually regrow some heart muscle, some cardiac muscle, and that is still being tested. But again, a really good way of using stem cells to support people and their medication or treatment. You may also be asked to consider the ethical considerations of using embryonic stem cells. Now, as we've previously discussed, embryonic stem cells are the most useful. They are pluripotent, which means they can turn into more cells than tissue stem cells. So that then makes them more useful for scientists and for therapeutic uses. However, that does come at a cost, because there are people who believe that these stem cells or these embryonic cells can become a life. Now, whether you agree with that or not, that clearly gives us some ethical problems in using them within science. As you can see here, there are 3 different places where we can get these embryonic stem cells. Each of them, although they may be useful for science, also come with an ethical consideration, and it has hampered some of the developments within stem cell usage. So it's definitely something to be aware of, and something again to have in your locker when asked a question about that. The key thing in terms of ethics is that some people consider that to be the destruction of a life or a potential life. So that's something to have in the locker. Now, I'm not going to go into a huge amount of detail here regarding specialised cell examples. You can see from this slide we've got quite a few options there, but within textbook, Scholar, Bite Size, you'll see a huge amount or array of specialised cells, not only in terms of the description, but equally pictures or diagrams of those. So again, you may be asked to identify the structure and the function, or the specialization and the function, and how they match up. So in multicellular organisms, there is more than one type of cell. We've got some examples here of both animal specialized cells and plant specialized cells. So they are specialized for their function, so they have a special shape or a special adaptation that makes them useful for that. And again, it's worthwhile noting some of those so that you've got some examples to talk about when it comes to tests and exams for specialized cell types. And the last part of us moving from cell biology into multicellular organisms is understanding the levels of organisation. So cells are the most basic of units of life, and those specialised cells that we talked about, they all perform the same function, and they group together to form a tissue. So for instance, muscle cells into muscle tissue. Those tissues then work together to form organs, and those organs then work together as a biological system. They perform then one or more functions within the body. And to show that as a clear example, cardiac muscle cells, so that's different from your structural muscle that is attached to your bones. Cardiac muscle obviously has to contract 24 hours a day, 7 days a week. That then forms cardiac muscle tissue. And as you know, then that will form the heart. And the heart isn't just muscle on its own. You have connective tissue, you have fat, you have valves, and other bits and pieces. So that is our organ. And that organ is part of the circulatory system. And that doesn't just operate on its own. The circulatory system is connected to the digestive system, the respiratory system, and other systems that form the human body or a multicellular organism. So it's worthwhile noting all those levels of organisation so that you know from basic cell up to systems and a body, that is the full level of organisation within multicellular organisms. And that wraps up our learning for Key Area 2.1, producing new cells. Right at the start, we talked about cell division being the source of all new cells, whether that's from the first moment of a fertilized egg developing into a fetus in the womb, or a young person growing to become an adult, or adults even just repairing themselves as they grow older. So growth and repair, that's the key reason we need cell division. Stem cells themselves are unspecialized cells. They can do two things. They can self-renew, so to create new stem cells, or they can specialize and into those specialized cells that have certain functions and therefore certain adaptations to fulfill that function. And as we've just talked about, cells are the most basic unit of life. They are grouped together into tissues. Tissues then form organs, and those organs work together to form a system, and many systems create that multicellular organism. So hopefully that's been helpful. If you have any queries, get in touch, and I'll speak to you again soon. Bye.